Dear students, here we shall be discussing the different aspects of meat spoilage caused by microorganisms, factors responsible for those, and the ways to control them. The spoilage of meat may be visible in terms of visual deterioration of color, formation of microbial colony, slimy layers on the surface, gas accumulation in packages, or false smelling compound generated over time. Like any other food products, there are some intrinsic and some extrinsic factors associated with spoilage of meat. The water activity of fresh meat is often above 0.99, indicating high availability of free water for microbes to grow. The pH range of 7.4 while the animal is living and coming down to as low as 5.4 after death of the animal is suitable for both bacteria and mold growth. Meat is nutritious for consumers, but it is as well as nutritious for microbes. In fact, meat extract powder is used in the nutrient media for growing microbes in the lab. This nutritious composition attracts spoilage microbes to grow very fast upon meat. The oxidation reduction potential also varies with changing pH and invites microbes to grow. The body temperature of animal when it is living is suitable for microbial growth, for which huge population of microbes survive or grow in the gastrointestinal tract of the body. However, as the animal dies, and if the temperature is not rapidly brought down, the microbes inside it can spoil the dead tissues very fast. And also, the fat content gets readily oxidized microbially or naturally, forming odorous, rancid compounds. Considering the extrinsic factors of meat spoilage, meat is mostly spoiled by microbes coming from the atmosphere, where animal is slaughtered and meat is cut. The moisture on the carcass surface and those in the atmosphere readily invites bacteria or mold to grow up. Even if the atmosphere is clean, contamination from dirty processing or cutting tools, hands, vessels, etc., comes in direct contact of the meat. The post-slaughter storage temperature, post-cutting storage temperature, and even the air inside the packages can spoil the meat. Although for people purchasing fresh slaughtered and cut meats from market for immediate consumption, these factors may not look serious, but for large-scale meat processing, for export quality production, and if you know, our India is one of the leading global exporters of meat, this microbial quality is a major issue for them. All these spoilage factors make meat one of the most vulnerable solid food. Strict safety protocols are necessary throughout the pre-slaughtering and slaughtering and packaging steps. So in this slide, we are showing a carcass of a pig. When the animal is killed, supply of oxygen from atmosphere immediately stops, and the inside environment becomes oxygenless, also called as anaerobic condition. The surface where the surface, however, remains in contact with air, and the aerobic organisms can grow here. Although microbes from the air cannot enter the interior of the surfaces easily, those adhering to the cutting devices surfaces can come in touch with the internal tissues while cutting, and some of those microbes will spoil, uh, and some of the microbes will be also spoilage causing anaerobic microbes, which will grow in the anaerobic environment inside the meat. So it is not necessary that all microorganisms coming in contact with the meat will survive. The aerobic ones may fail to survive because the condition is anaerobic, but the anaerobic ones will survive and cause the spoilage of the meat from the inside. So thereby spoilage on surface will be by aerobic microbes and spoilage inside the muscles will be by anaerobic microbes. These microbes are mostly bacteria and some are molds. Some cause disease in consumers, some simply affect the meat's quality while growing over it. Now, you need to remember the microbes responsible for spoilage and pathogenicity that has been put on the slide. The different types of quality deterioration by aerobic and anaerobic microspecies are given in this figure. You can see that the effects of aerobic microbes are higher than non-aerobic or the anaerobic. But the most dreaded microbe, Clostridium, and its botulinum species can grow in meat, and it's an anaerobic putrefactive, that is protein degrading bacteria. So Clostridium botulinum can grow in meat, and it's an anaerobic protein degrading, which is called putrefactive bacteria. And these are the various spoilage types caused by aerobic bacteria in stored meat. The formation of slime on the surface, pigmentation on surface, and off-flavor generation on the surface of meat are characteristic spoilages. Off-flavor is generally rancidity, which is oxidation of fat by lipolytic bacteria or by natural means. But it can be also due to degradation of amino acids to amines and ammonia by some bacterial species. Anaerobic spoilage can be putrefaction by putrefactive anaerobes, which are a group of anaerobic microbes causing degradation of protein and forming foul-smelling compounds in meat. Or it can be souring caused by lactic acid bacterial species, causing additional formation of lactic acid apart from the naturally occurring post-mortem formation of lactic acid. Some lactic acid bacteria can again cause lactic acid fermentation, increasing the amount of lactic acid in the meat, resulting in a sour tasting meat. So souring and putrefaction are the two characteristics of anaerobic spoilage bacteria. Safety measures while handling meat are therefore essential. If the factors for spoilage, both intrinsic and extrinsic, are controlled, spoilage can be also controlled. However, all intrinsic factors cannot be controlled. As we need our purchased meat to be juicy, and the seller cannot afford any loss due to difference in weight due to drying, the water activity cannot be reduced. Although curing with salt reduces it to an extent, cured meat is not often considered equivalent to raw meat. The nutritional composition can be compromised, and it is not ideally possible to reduce portions of the nutritional components extraneously, so it's not even a question. The body temperature of living animal will remain what it is. We can deal with the dead carcass only. So the temperature of the living organism before killing it we cannot control. After killing, of course, we can go for freezing, frozen storage. That can be done, but during when it is living, this intrinsic factor you cannot control. The gastrointestinal tract will remain there until slaughtering. The fat content cannot be changed or reduced. However, we can regulate pH change or pH level, as pH is associated with the metabolic status of an animal just before death, and accordingly the oxidation reduction potential can be also partially regulated. How the pH is associated with the metabolic condition of the animal, that I'll be discussing in the next slide. Although very less can be done to control intrinsic factors, every extrinsic factor can be regulated in a fully equipped slaughterhouse. Therefore, safe handling encompasses adequate handling of animals before death, during slaughtering and cutting, keeping the process environment hygienic, and, and practicing micro-free packaging practices. So, discussing the relation between the pH and the metabolic state of an animal. So, before death, if an animal is exercised followed by giving it sufficient rest. Suppose the animal is fully exercised, after that it has been given sufficient rest, the glycogen is used up to a great extent during the exercising, and during the rest it will neutralize. So due to unavailability of the glycogen in the tissues, the extent of glycolysis will be limited after that. Hence, formation of lactic acid will be lower and pH will be higher. The final meat pH will be on a higher side. But if the animal is killed under stress, that is, if it is not fed well, agitated during killing, not in a good state of health, and hence is restless, then high rate of post-mortem glycolysis will occur and pH will drop rapidly due to lactic acid generation is in huge quantity. In low pH meat, muscles are rapidly degenerated and it will release interstitial water. Those interstitial water are bound to the muscle fibers. So as the muscle fibers will shrink, those bound water or interstitial water will be released out. As the bound water turns into free water now, the water activity will rise and the microbial spoilage will also become rapid because the water activity is has been enhanced. As the meat is thawed after freezing, the oozing out of water from the meat will be also higher in case of low pH meat, as the amount of free water is higher, and which is not a sign of a good quality meat. So this is the difference between high pH and low pH meat. Generally, high pH meat is favored over low pH meat for has a better quality meat. The difference in cooking characteristics of high and low pH meat have been discussed in the previous part of the lecture series. So in the you can go to the previous lecture, you will find the difference in cooking properties of low pH and high pH meats. The photographs here show a wounded, dying deer under stress, and below is a cattle in rest being bolted to slaughter. The wounded deer will be producing low pH meat, while a high pH meat can be expected from the bolted cattle, as the bolted cattle is in rest during its death and its energy metabolism is not highly functional. But in case of the deer which is running, there will be high metabolism of glycogen, and the liberated glucose will keep turning into lactic acid after that, and there will be formation of low pH meat. During slaughtering, the following sources of contamination must be avoided. Animal fur or animal hair and skin surface are natural sources of microbes. So naturally the skin and hair, the fur will be having some microbes from the atmosphere. So they should not come in contact with the meat. So the surface should be sanitized, or preferably hair should be shaved or removed before slaughtering or before cutting of the meat. Number second is blood. And blood is a medium where microbes like to grow. It's a liquid, it has all the nutritional resources. Microbes would like to grow in the blood. So the lesser the blood in the carcass, safer is the meat. So you have to get rid of the blood. So blood from animal is often drained during killing in the halal method, which can be related to the microbial safety of the stored meat. The viscera of the or the gastrointestinal tract is a hub of microbes as well as hydrolytic enzymes. Therefore, removal of viscera should be immediately followed after slaughtering, deskinning of the animal. The processing tools should be sterilized before and after operation of slaughter or cutting. The knife should be always kept sharpened and stain free. Now, as you know by now that safety is globally associated with positive quality of product including meat, and a business delivering quality product and following safety standards can only sustain longer. It is said that the light of knowledge will enlighten the world, making it better place to live. So the knowledge of meat science, the knowledge of the slaughtering industry, the meat industry is important, and gradually it will enlighten the world of meat industry globally. Thank you very much. Take care.